Okay.
Perfect.
Okay, great. Thank you. I do want to just make a comment that I am not a doctor. I'm not a DVM. I was just a director in the program at AstraZeneca. So today's presentation was put together by the North American 3Rs Collaborative. And before we go into any specifics of the presentation, I want to briefly touch upon the organization and why we decided to focus on refined mouse handling. Our mission is to advance science, innovation, and research animal welfare through collaborative 3Rs efforts. We do this by facilitating diverse collaborations, and this is just our board and leadership team. We're comprised of end users such as pharma, contract research organizations, commercial suppliers, biotech, government academics, and other nonprofits. This collaborative effort makes us a powerful force to forward the 3Rs. And I do want to apologize, I missed the first slide, which I do want to mention that the presentation would not be possible without the support of Megan LaFollette, the program manager for the North American 3Rs Collaborative, Dr. Mark Prescott, who's the Director of Policy and Outreach for the NC3Rs organization, and Dr. Liz Nunemaker, the Director of Animal Welfare for Charles River Laboratories. I've been involved with the North American 3Rs Collaborative refinement team for about 2 years, and I'm very excited to see the progress that has been made in the US on this initiative. Megan is managing the program and doing an exceptional job at bringing this information to the North American lab animal community. I would also like to clarify that refined handling has a few different names in the literature. It's also called non-aversive handling or low stress handling, but these are all generally referring to either handling the mouse with a tunnel or using cupped hands. So the North American 3Rs Collaborative strategy is to identify key evidence-based 3Rs approaches, address barriers, and promote them as everyday practice. Our goal is to choose initiatives that have strong evidence, will make a big impact, and can ultimately be practical. So the NA3RC currently has 6 main initiatives: rodent health monitoring, refinement, translational biomarkers, microphysiological systems, 3R certification course, and compassion fatigue. And today we're going to focus on our efforts of the refinement group. In our view, refined mouse handling has strong evidence and can make a big impact and ultimately is practical. There have been numerous publications over the past 12 years contributing to the growing body of evidence to support refined handling. Selected references are seen here at the bottom of the slide. In general, refined handling techniques lead to animals that are more comfortable being handled. They have increased voluntary interactions with their handlers. They're less likely to try to escape, and they experience less stress associated with handling. These animals also exhibit lower levels of anxiety, which has been demonstrated through multiple behavioral tests. They have a decreased physiological stress response, a stronger reward response, exhibit fewer depression-like behaviors, and have decreased variability in behavioral and drug studies. The literature consistently demonstrates that picking up mice by the tail has negative effects compared with refined handling methods. Not only does refined handling decrease animal stress and anxiety, these effects have lasting impact on the animals that are restrained for procedures. In this figure on the right of the slide, we see the percentage of time animals spend voluntarily interacting with the handler before and after scruff restraint for 3 different handling methods: tail, tunnel, and cup handling. These last 2 represent refined handling methods. Animals that were tail handled spent less than 20% of the time interacting with their handler, and this dropped to 10% after scruff restraint. Animals typically handled with tunnels or cupped hands interacted with their handler 50 and 30% respectively. but there was no drop after the scruff restraint. It appears that only mice that are picked up by their tail avoid handlers after scruff restraint. This experiment has been repeated multiple times for restraint handling, including restraint by the tail, blood collection, and anesthesia, and each time we see the same result. Animals continue to interact with handlers when refined techniques are used for routine handling. Collectively, these results suggest that it's really the picking up the animals by the tail that is truly aversive for our mice, and that refined handling can have a lasting impact on animal well-being. So refined handling is practical even for large institutions. Non-aversive handling has been implemented exclusively by over 60% of UK institutions. At least 2 large animal facilities in the US have made the switch. They report it takes no more time after proper training is implemented. And people say that once they've switched, that they'd never go back. I definitely agree with that statement. We have done that at our facility and they would never go back. So today you're going to hear from myself and Liz on our experience in the evidence, impact, and practicality of transitioning to refined handling. I'm going to give the next presentation, and the presentation by Liz was recorded previously. She was unable to do this live virtually at this time. So until recently, I was the director of the Animal Sciences and Technologies program at AstraZeneca. I'll refer to the program as AST. I retired in April after 18 years with MedImmune/AstraZeneca and 39 years in the laboratory animal sciences field. I was given permission by AstraZeneca to present this information that was generated at our Gaithersburg site. And I will probably slip back into saying we and our site a lot because old habits are really hard to break. So the animal care program at AstraZeneca in Gaithersburg is about a 20,000 square feet animal facility, which houses rodents only. AstraZeneca worldwide has 4 sites with animal facilities, 1 in Sweden, 1 in the UK, and 2 in the US, the other being in Boston. The Gaithersburg site is the only one that's USDA registered due to occasionally housing cotton rats. The caging on site is Technoplast Green Line and Slimline Plus caging. Generally, they house 5 mice per cage in the green line and 3 mice per cage in the blue line. There is some limited special housing where they do automated feed measurement and the animals are individually housed, and there's some caging that's positive pressure micro-isolator caging for germ-free mice. The areas of research include oncology, cardiovascular and metabolic disease, respiratory disease, and infectious disease, and they're just starting back to do vaccine development. In the past few years, a new program to evaluate the effects on the microbiome have on the treatments for metabolic disease and cancer has been added. And those are the mice that are housed in the positive pressure micro-isolator caging. The animal care program comprises a group of husbandry staff that are responsible for changing cages, receiving animals, doing the cage wash functions, facility maintenance, and providing some limited technical support. The other part of the staff is the technical staff that support the therapy area scientists. And they do procedures such as dosing via various routes, blood collection, surgery support, imaging support, study data collection, and they train the animal care staff and the scientists. So how did we start in this process? Well, in 2019, the global AST leadership team decided to set a goal for all of the sites that included implementing alternative methods for mouse handling as our standard practice. So in Gaithersburg, the team began reviewing the UK documentation and training materials that they were provided by the UK colleagues who had been implementing these methods for several years. I think they began in 2018. And because this was on our group goals, we had the financial commitment to order clear tunnels for every cage in the animal facility. In the UK, they were able to reach out to Jane Hurst's team to get them some help to get them started and provide training. But in the US, we actually got a bit lucky in that area as well. For the past 4 to 5 years, AWIC has asked us if we could provide a tour of our animal facility for the UK AWIC Fellowship Award winner. And the UK Fellow generally spends the first week of their fellowship program in the Maryland, DC area, because there are a lot of different types of animal facilities in a relatively close geographic area. So in 2019, the fellow was John Waters, and John worked in Jane Hurst's lab, and he was an author on a number of publications on non-aversive mouse handling methods. So we were thrilled to hear that he was going to come visit our site. I contacted him in advance and asked him if he would give our staff a presentation about mouse handling, and then spend a little time with them, working with them, and providing them some tips and tricks on how to improve the process and how to make it easier. This additional encouragement was just what everyone needed. He was incredibly helpful, which really increased the motivation of the team to fully integrate this into our animal care program. And by the end of 2019, all the ASD staff were well trained in these handling methods, and we initiated the requirement that no mouse would be picked up by the tail. So as we got comfortable, as ASD staff got comfortable with these handling methods, they started training the scientific staff as well. And several PIs from each therapeutic area were selected to be champions on this project so they could help other members of their teams. But when the pandemic started, we did decrease some of the animal numbers in the facility, but we did continue our research. It did actually help in this process because there were fewer PIs in the animal facility, so the ASD team could watch them and then help them if they were struggling with implementing this process of handling the animals using a tunnel or cupping method. In July of 2020, as we began to allow more PIs into the animal facility, we sent messages out to all of the animal facility users that picking up the mice by the tail was no longer an acceptable practice. And if they needed help in handling the mice, the AST team was available to help them. And at this point, the AST management staff tried to track concerns or issues that the PIs brought up with this new process. So some of the common concerns that we had, the PIs involved with the microbiome projects were concerned about potential contamination from the tunnels in their cages if they weren't washed or autoclaved properly. So it was decided that no tunnels would be used in those cages and the cupping method would only be used. Some of the mice on the diabetes and obesity studies became too fat for the tunnels. And we found that they adapted to cupping very well. So we decided just not to use tunnels in those cages as well. We did have some issues with water leaks from tunnel placement in the cages. Since 95% of the cages had automatic watering, this issue needed to be managed very quickly. When changing the cages, the animal husbandry staff always placed the tunnel on the side of the cage parallel to the side of the cage. But the investigators routinely would just plunk the tunnel back into the cage when they were finished handling the animals. And then the mice were able to push that tunnel up against the stem of the water valve and it would start leaking. So we sent out an email to all the animal facility users about tunnel placement using these pictures and explaining how to do it properly. But in 2021, we switched to a shielded valve in all of the cages. So we replaced valves in about 4,000 mouse cages to a valve that has a recessed stem. And this almost completely alleviated the problem with leaking cages. So as this was a new procedure that affected every in vivo user, continuing continuous training was needed. And every 2 years, all the animal facility users were required to do a refresher training, a humane science refresher training. So that was done in November of 2020. And we spent a bit of time discussing implementing the alternative methods. And we emphasized that no mice were to be picked up by the tail. We generated some training videos, and we provided them that, and we provided them the literature that was out there explaining why this was necessary. All newly hired investigators have to go through a humane science training and animal facility training. And this includes discussion on mouse handling methods. And we went to the next step, requiring a training session with one of the AST trainers to demonstrate how to handle mice properly, and then to show the trainer that the investigator knew how to do that. And that is then recorded in their training records. So I have a couple of videos next to show you just what we did on our training videos. Let's see, hopefully this works. Okay, so some of the questions we had about not picking mice up by the tail was, well, even if we're going to pick them up with a tunnel, we're going to have to handle them by the tail to restrain them for certain procedures. And we explained to them it wasn't the act of touching the tail, it was the act of lifting that mouse from the cage with the tail. And generally what you're seeing here is what the scientists would do is go in and get all their animals out of the cage, put them on top of the wire bar feeder, and then get them prepared for whatever procedure that they were going to do. So this just was to show them how easy it was to get the mice out of the cage, and then show them how they needed to be handled once they were put on top of that wire feeder. So this person is just doing a tumor measurement on the mice. So the next video shows someone taking the mice directly out of the shipping crate and examining them, and then putting them into their new home cage. And we use this example to show them that here are some animals that have never seen a tunnel before. But if you know what you're doing and work efficiently, it's very easy to get the mice into the tunnel and then into the cage. And you see the technician getting the mouse, slightly restraining them by the tail in the back of the tunnel just to sex them. And then this is just a video on using the cupping method to remove the animals from the inside of the cage. And these mice had obviously been handled before, because they very quickly got onto the technician's hand and were easily picked up to be put on top of the feeder and then to be handled for an injection procedure. And again, they're restraining them with the tail on top of the feeder, and then just putting them back into the cage. So we also thought of some other ways to improve our mouse welfare for our program. And we asked 2 of our vendors about placing tunnels in cages of mice on our breeding projects. So those mice would be at least familiar with a tunnel in their cage. They would not ship the mice to us with tunnels in the shipping container. They were concerned about that, but they would put the tunnels in the cages. Early on, our animal husbandry staff asked if there were other methods we could use to move the mice, such as using a feed scoop to move the mouse from the dirty cage to the clean cage. They thought it might be easier. And we wanted to encourage that type of thinking, so we let them try out the method. They found themselves that it wasn't any faster than using the tunnel, and they felt that too much dirty bedding was getting moved from the dirty cage to the clean cage. So they decided that wasn't effective. They would just continue to use a tunnel or the cupping method. So after about a year of implementation of the practice, we decided to send out a survey to all the animal facility users and ask them about this new refined mouse handling method. We received approximately 50% of the responses from routine animal facility users, and the responses were from 75% of the research scientists and 25% of the AST staff. Almost all indicated that they were moderately or very familiar with non-aversive mouse handling techniques. And this was good. It demonstrated that we got through to them and they remembered it from the training. 75% of them said that they use these methods more than 50% of the time, and only 25% said they used it less than 50% of the time. So we knew that there was still a little bit of work that needed to be done. Only 2 people thought it was not useful at all. But the survey did give us some insights on how to improve the program and the training. And an updated survey has been created, and it's, I think, been sent out. I'm not exactly sure. But this will be discussed at the ALAS meeting in October. So there were several misconceptions that remain. Again, they felt that mice still needed to be handled by the tail for tail IV injection. What difference does it make picking them up? And we explained that they're being handled by the tunnel, and you're not lifting that mouse by the tail. That's really what's causing that stress. They said, well, the new mice are too fast. It takes too long to get them in the tunnel. Hence, that's why we did the video on getting mice directly out of the shipping container into the tunnel and demonstrating to them that could be done. Some people indicated that, oh, it would take too much time if you were doing procedures with a lot of mice, like randomization. And we stressed that the additional time spent up front on this process may be slightly greater, but the time savings was not an acceptable reason when it came to the welfare of the animals. Some folks who had experience with certain strains of mice believed that their mice were just too fast or too jumpy to be directed into the tunnel. And we recognized that this was not true because the husbandry team were easily able to do this with the same mice. In one animal holding room, we had mice on an oncology study that the AST scientists and the husbandry technicians could easily identify which PI was handling their mice with the tunnel and which were not. The same strain of mouse from the good PI were very calm and easily handled. And the other set of mice from the PI that wasn't following processes were still difficult to handle. I do especially like the comment that tunnel handling hasn't been validated for my models. We had very few sensitive models at AstraZeneca, and we would counter to them whether they were aware of how the stress on the animals affects their models. And they generally didn't know about that. So overall, we believe we had good success in implementing the practice in our program. We received really good feedback from ALAC in 2021 during our site visit that implementing this practice showed our commitment to improvements in animal welfare. This process involved a team of people from across the organization. Our Gaithersburg team supported the training of the AST team and the research scientists, and we had about 100 to 120 research scientists that they needed to train. I would like to give a quick plug for a roundtable session that the North American 3Rs Collaborative is doing at the ALS meeting in October on mouse refined handling methods. The NA3RC sent out a survey in 2020 about refined mouse handling, and we will be discussing those survey results. We hope to have a robust discussion with folks who have already implemented this practice, and those who need or want more information about it, we'd like to be able to help them implement it as well.